Good morning, one and all. Welcome to the second day of this International Symposium on Bioinformatics. I'm very much delighted to give the brief biodata of Professor Gajendra Raghava. Professor Raghava received his MTech from IIT Delhi in 1986 and PhD from Punjab University in Tech Chandigarh in 1996. He worked as postdoctoral fellow at Oxford University, UK, bioinformatics specialist at UMS, USA, and visiting professor at postdoc South Korea in the year 2004. He worked around 30 years at different scientist positions in the field of bioinformatics at CSR Institute of Microbiology Technology, Chandigarh. Presently, he is working as professor at IIIT Delhi in the field of computational biology. He has published more than 250 research papers in reputed journals. He got Thomson Reuters Research Excellence India Research Front Awards in 2009. Because of his two papers, he ranked in the category of highly cited papers in the world. In 2014, he has been listed in the world's most influential scientific minds in the field of computer science. His group developed more than 250 in silico products, which is highest contribution by a single group in the world. He is a strong supporter of open source software, web servers, all services developed at his group are free for academic use. These web-based services are heavily used worldwide, more than 150,000 hits per day. He got numerous awards and recognitions. Few have been listed below. So I'm going to read one of Shanti Swarup Bhatnagar Award in Biological Science in the year 2008, National Bioscience Award for Career Development in the year 2006, J.C. Bose National Fellowship from the year 2010 to 20 by DST, Fellow of National Academy of Science, Fellow of Indian Academy of Science, and he is also the Fellow of Indian National Science Academy. With this, I request Professor Gajendra Raghava to deliver his talk. Sir, please.
Sir, please unmute yourself.
Hi.
Hi, sir. Sir, I had enabled the screen sharing. Please share your PPT and.
Just hold.
Okay, sir.
It is showing disabled sharing actually. You have to allow me to share.
Sir, please share, sir. Already we had enabled you.
Okay. Is it shared?
Yeah, we can see this. We can see it.
Okay.
Anil, tell professor to have this full screen display.
Yes, sir.
So tell me when I should to start.
So you can start now, sir.
All right. So today talk is about the computer-aided immunotherapy to fight against emerging pathogens. Right. So most of the people in bioinformatics, they mainly focused on the drug discovery, but our group, one of the major focus is on immunotherapy or vaccines. So I will go through each and every step. Let's say just come straight to understand the immune system. Why we got the disease? There are two types of disease. One is through the attack by the pathogens. Maybe they are called microbes basically. Bacteria, fungus, parasites, virus. These are some of the examples. Okay. Or the disease may be due to the disorder of our own own body system basically. Okay. So I'm talking about disease caused by pathogens. Maybe it's due to bacteria, virus, this sort of thing. Before I talk further or explain the concept further, it is important to understand the defense system actually, because without doing, without understanding biology or immune system, you cannot develop the tools for bioinformatics. That's the most important part. So let's say when any enemy or disease causing agent attack a human, so what will happen? There will be two types of defense. First is non-specific defense, or we call innate immunity. Innate immunity will come into the picture, and they will release certain chemicals, certain toxic materials, so that they will kill the disease-causing agent, bacteria, fungus, whatever, pathogen basically. And these pathogens are recognized by the receptors actually, that is called pattern recognizing receptor, PRRP, and main class of the PRRP is toll-like receptors basically. Okay. So they recognize pathogen associated molecular patterns actually, because each this pathogen, bacteria, fungus, they have certain type of pattern on the surface actually, and this is recognized by the innate immune system, and they give the signal to the body, look, there is an enemy, and same time they release certain chemicals to provide the first line of defense. So this is the one part of the system, innate immunity. There is a specific defense. Still there is some virus or bacteria which crosses this innate immunity actually, which bypass actually. So then there is a next system that is called specific defense system or adaptive immunity. In case of the adaptive immunity, then there is a two part, humoral immunity and cellular immunity. In humoral immunity, there is a B cell basically, which release the antibodies which can directly neutralize the pathogen. There is another category that is called cell-mediated immunity, where they have the T helper cell and cytotoxic T cells basically. These are the two categories, and these cytotoxic T cells, they release certain chemicals and they kill the infected cell, where the T helper cell activate both arm, means B cell as well as cytotoxic T cells. So let's say for as a bioinformatics, what is challenge? Challenge is initially the vaccines are basically based on the whole pathogen. They take the whole pathogen, they kill it or annihilate it and give to the body. So what will happen? Then there will be immune system will thought that enemy is there and then defense will be prepared. During the defense preparation, they will generate the memory cells. They may be called the So I was talking about the B cell and T cell memory generations. Now I will go to the next part. Okay, so first we have to understand that how the antigen is processed, how a pathogen is processed basically. Let's say if bacteria or virus. Okay, so if any pathogen goes inside the cell actually, what will happen actually? Let's say this is the example that pathogen is here, which is a disease causing pathogen, and what will happen? Because these pathogens, these are the living organisms, they also release certain type of proteins basically. These are called the secretory proteins. Okay, these secretory proteins are not the same as our human proteins. So we have the proteasome system in our body. This proteasome recognizes foreign proteins, and once they recognize it, they chop it into the pieces actually. Pieces in the means term of, let's say, small peptides, 9 amino acid peptide, 10 amino acid peptide, whatever is there, they chop it depending on the enzyme cleavage site basically. Now question is basically how to predict that what type of fragment will be generated from a pathogen protein actually. So there are a number of softwares have been developed in the past. Our group also developed a software called p-cleavage. This is our group software where we predict whether this protein will be cut or not. If it is cut, then from where it will cut, means what fragment will be generated from a protein. So this is the one part, you get the peptide corresponding to a protein. The next part is there is a ER system, endoplasmic rectum is there, it has to go there actually. Okay, then there is a TAP proteins are there which work as a receptor. So they only the peptide can go inside the ER which can bind to the TAP actually. So we need to predict, we need to develop a method which can predict whether a given peptide will bind to the TAP or not. So that's the second condition. So number of groups have developed methods. Our group developed the method called TAP-PRED, and this TAP-PRED allow to predict whether a peptide will go inside the ER system or not. Once the PR is in this peptide, selected peptides is inside the ER, there is a MSC class 1 molecule, or we call HLA1. HLA1. There are different alleles of HLA1. Okay. Then some of the peptide will bind to HLA, not all. The question is which peptide will bind to the HLA. That's a challenge. So we developed the method to predict which peptide will bind to which HLA allele actually. So that method is called PRRDB from our group, and there are a number of other groups who are also involved in this business. They also develop the tools actually. These tools are available. Once a peptide of a pathogen bind, then it will take the HLA molecule and will take the bound peptide to cell surface. It means ideally the pathogen antigenic region is ultimately taken by the HLA to the cell surface. Here is a CD8 cells are there, or called cytotoxic T cells. These are cells which are monitoring the system if there is any infection in any cell. Once they find out that one MSC molecule or HLA molecule is having the bounded peptide, then they will bound to it, T cell receptor will bind to it and will recognize that cell is infected. So then what will be the what will happen? Then CD8 cell will release certain type of cytokines, and these cytokines will kill the infected cell. So that's a mechanism for killing the infected cells from the body, just to clear the infected cells from the body. So here we have a method for predicting cytotoxic T cell for MHC binders. Okay. So here is requirement of immunoinformatics. For each and every step, we need to develop the method to predict the right candidate. Because if you do the same thing with the experimental techniques, it will take a long time actually. Okay, so practically it's not possible. So with the help of the computer, you can find out which peptide will activate our CD8 cells. So there is two actions here. CD8 cell will release certain cytokines and will kill the infected cell, as well as the cell which recognize this part will generate the memory B cells, memory T cells, so that in future, if there is an infection, then sufficient number of CD8 cells which can recognize this pathogen is there in the system. That's the basically concept of the vaccine. So whole life you have the large number of T cells which recognize a particular pathogen. Okay. This is another concept. When your pathogen is outside the cell, actually. In this situation, pathogen is not inside. Here is a pathogen. Okay. So they are releasing a certain type of proteins only, actually. They are outside the cell and releasing a certain type of proteins, basically. These proteins will be engulfed by your antigen presenting cells. These antigen presenting cells will take this engulfed protein or antigen into the lysosome, actually. Lysosome is full of enzymes, basically. They will chop it into the different pieces. So there will be protein will be cut into the different peptides, actually. Once they are cut into peptides, then they will go bind to MHC class 2 molecule. Please remember this thing. Earlier I was talking about MHC class 1 or HLA1. Here I am talking about the MHC class 2 or HLA2. Okay. HLA and MHC class 2 is same thing. Only thing in case of the human, we call the human leukocyte antigen, actually. That's why we call HLA. Generally the word is MHC, major histolytic complex. Okay. Then it will be again presented on cell surface. This is a MHC molecule, this part is a MHC molecule, and this is a bounded peptide here. Okay, so this will be recognized by CD8 cells. Please remember, earlier it was CD8 cell. Here it is CD4 cell. So the concept is, if peptide is presented by MHC class 2 molecule, then CD8 cell will recognize it. If it is presented by class 1 molecule or HLA1 molecule, then it will be recognized by CD8 cell. That's a major fundamental difference. Now, question is, can we predict which peptide will bind to the MHC? So we have the software MHC, this ProPred, which will help you. There is other softwares also from our group, MHC, 2Pred and HLA Pred. They help to predict whether a certain peptide will bind to the particular MHC class 2 allele or not. Now, CD8 cell, or this is called the T helper cells, actually. They also have their branch TH1, TH2, and they release certain type of cytokines, actually. Some of them are basically interferon gamma, interleukin 4, like this, actually. Okay. So these are the methods where our methods simply predict this IFN epitope, predict whether it will release or induce interferon gamma or not, actually. These are particular type of cytokines, actually. So it will not only tell that it's a MHC binder, it will also tell whether it will release the interferon gamma or IL-4, actually. Because sometimes you need the specific type of cytokines in order to handle the particular type of disease. Okay, so there is another software called ProPred, actually, help to find out the MHC class 2 binders. And beauty of this software is that it helps you to identify the region where the most of the MHC class 2 alleles will bind. That's important to design a vaccine which can work for the wide range of population, actually, rather than for the specific class of population, actually. So that is called promiscuous MHC binders or epitopes. So I'm not going in the detail. These are the different methods. Similarly, there is a methods we have developed for predicting the B cell epitope. It may be conformational B cell epitope, it may be linear B cell epitope. So if you are interested, you can use them. So it started long time back, 20 years back. We realized that there is no database for the B cell epitopes actually. So we collected and compiled this information, and then we created a database. This is important part. That's what we changed the trend actually. Most of the cases, if you see the history of Indian science actually, that most of the time database is created by the big countries like US, UK, and other countries. We are mostly users of them actually. That gives a lot of disadvantage to us actually. But what we did actually, we created the databases, which are the primary databases. So anybody who want to work in the field, they can use this database. That's the important part actually. So we have the source of the data. And we evaluate the first instance the all existing B cell epitope prediction methods, and then we realized the existing methods at the time in 2003 that most of the methods they have the poor performance, because 50% you can get by chance and they are getting maximum 57%. Then we combine all these physiochemical properties and then we reach up to 58.7%. That was the maximum achieved by any method actually. Okay. There is a first method we developed using the machine learning technique for B cell epitope prediction actually. Before that, nobody knows how to implement the machine learning techniques or neural network in predicting the B cell epitope. That was a major challenge actually. So we make certain assumptions, and based on these assumptions, we develop the method for predicting B cell epitope, and we achieve the accuracy something like nearly 66% actually. Though it is not very high accuracy, but seeing the other methods, physicochemical property based method, this is much better than the existing methods actually. And this method later become the standard in the field. Everybody is used for B cell epitope prediction. Same we have the tool for conformational B cell epitope, but this method is designed from the sequences only. Most of the method they require the structure in order to predict the conformational B cell epitope. But we develop the method which need only the sequence, and from sequence we predict the conformational epitopes. Same way, this is an updated version of the ABCPRED, latest version. This is another interesting paper or software we have. Most of the methods in the literature, they only predict whether it's a B cell epitope or not, means the peptide which can induce the B cell or not. But they don't tell what type of antibody will release actually. This method tells that whether the peptide will release IgG type of antibodies, IgE type of antibodies, or IgA type of antibodies. So that is a further classification. The type of antibodies will be released by a peptide actually. Okay. This is for the innate immune system, because the innate immune system is an important part we have to understand. So we created the database for the pattern recognizing receptors basically. This is available. This is another part, innate immune system. We can identify the peptide-based vaccine adjuvants. Peptide-based vaccine adjuvants. So we have developed the method for peptide-based vaccine adjuvants, nucleotide-based vaccine adjuvants, and RNA-based vaccine adjuvants. So these are the different type of adjuvants we develop, and this is based on the TLR recognition. Because this is important. Most of the bioinformaticians thought that if they know the computer programming, then the problem will solve. No. You have to understand immunology or biology in detail in order to develop any method. Then you can develop the methods for that problem basically. So here is a method where they help to design the vaccine adjuvants actually. That is called simply in the drugs salts actually. Okay, so here is a peptide or RNA or DNA based vaccine adjuvants. Because nowadays there is a problem of allergy actually. A lot of genetically modified foods are there actually. Okay, we don't know whether the particular protein which is introduced in the market is allergic or not actually. So this method predict whether a given protein is allergenic or not, as well as IgE epitope. IgE epitope is directly related to the allergy actually. So this is a summary of the tools we developed in our group for different purposes. Let's say T cell epitope. These are some of the important tools. I'm not covering all the tools from our group, only covering few tools. If you are interested in a B cell, these are some tools. If you are interested in the vaccine adjuvants, basically, then these are the tools actually. So these are the different type of tools which is available from our group. You can use them actually. Beside this, because in vaccine you are normally predicting the epitope or antigenic region which can serve as a immunotherapy or as a vaccine candidate. But there is another thing actually. What is the half-life of the peptide? That's another important part basically. So we have the developed the software which can predict the half-life of the peptides in given let's say in different body fluids basically, in blood actually. As well as you have to predict toxicity of these peptide or protein because they may be toxic to the body actually. So we have the software called Toxin Pride actually, which can help you to predict the toxicity. Believe me, this year, due to the coronavirus and other things, the Toxin Pride, the server, is heavily used, cited more than 170 times actually in last few months basically. Then there is another type of toxicity, that's a hemolytic peptide identity, means they lyse the RBC cells actually, because there are different type of cells. These peptides, they can lyse the RBC. So we cannot use them as a vaccine. So there should be some server which can help you to predict. So this is for toxicity prediction. Then these are the examples, some portal we have given where you can work. Let's say this portal is for tuberculosis actually. If you want to design the tuberculosis vaccine against any strain, including new strain, because sequencing new strain is not a difficult task, then this portal will help you to identify the vaccine candidates. Same time, let's say a few years back, Ebola virus was there actually. So we developed this platform just to help the biologists. We predicted the tools, we predicted the potential vaccine candidate, drug candidate, diagnostic candidates, so that you can use them from our platform actually. Same way we did for the nowadays concept of the vaccine. I'm not going into the detail because these are the different type of trials actually. Okay, or some of them are already in the market. So we point out that there are chances that the vaccines may have some side effects actually. So be aware about them actually. And we also suggest the possible solutions to check their toxicity and the region of side effects actually. This opinion paper we published a few months back, and we already work on the one cytokine which is basically responsible is IL-6. Because IL-6 is responsible for interferon gamma. We have already developed the method earlier actually. So recently we developed the method for IL-6 also, so that it will predict whether it will induce the IL-6 or not actually. So this is a server. If you are interested, you can use it. Believe me, we have more than 250 web servers which are available to the public. So if you are interested, you can use them actually. All are free to the public. Okay, the type of information is stored here. This is a web page. You can use it if you are interested in the coronavirus actually, and you can get all type of informations. What is a genome, genomics, related informations, what type of genomes have been sequenced. So what we have done actually, we have just collected and compiled the information from the literature. So we are not there generating the data. We are just collecting and compiling the information from the literature so that user can use it actually. Same way there is a this menu allow diagnosis. Let's say what type of diagnostic tools are there. We predicted the different type of primers actually for predicting the this disease actually. And then we have developed the immunotherapy tools, the possible peptides, which can serve as a vaccine candidate. Same, there is a drug design software, structures of these targets, everything is given here. So the overall aim here actually, we are collecting, compiling, and predicting the information related to the coronavirus, so that anybody who is working in this area may get the help from our portal. We don't need any credit or anything. Anybody can use them actually. They are all open actually. Thank you very much. I think I am within the time actually. Okay, thank you.
Thank you very much, sir. That was such a wonderful talk.